Protein molecules consist of thousands of atoms. Their size range from less than 50 amino acids in pancreatic polypeptide to several thousand in large photosystem complexes. Protein primary structure, amino acid sequence, determines its secondary, tertiary, and quaternary structure. We compare protein amino acid sequences to identify common structural elements in different proteins and sequence motifs responsible for interactions with ligands. Sequence comparison gives us invaluable source of experimental design and protein engineering. Proteins with high sequence homology possess similar three-dimensional structure. However, low sequence homology also can result in the same fold if the key core positions, determinants of fold, are conserved. One sequence stretch in a protein can be homologous to several different sites of another protein. This happens if protein structure has repeats of tertiary structure elements, such as a pair of alpha helices, metal binding site, or a collection of beta strands. Proteins with no homology can have short stretches of homologous sequences, which very often indicate presence of common ligands, phosphorylation sites, or other features. Sequence alignment methods compare amino acid sequence of two or more proteins and identify identical or homologous stretches. The score is calculated to show number of identical or homologous positions in the two proteins. If sequence homology of the two proteins is high, their tertiary structure is very similar. If sequence homology is low, there may be ambiguities in assigning tertiary structure based on amino acid sequence. Since protein secondary structure elements interact with each other in a highly specific manner, amino acid of one secondary structure element correlates with amino acid sequence of another secondary structure element. Amino acid combinations at helix-helix interfaces are related to each other. If we consider amino acid sequence not of each individual helix, but pairs of alpha helices, sequence homology becomes more reliable source of tertiary structure similarities. Structure of proteins that has low homology of the linear sequence stretch, but higher homology of the amino acid combination at helix-helix interface is predicted more reliably.